Sindika, Director in National University, and today I am going to talk about some preliminary results about the in vitro degradation, and which is obtained from paradioxanone, L-lactide, and polyethylene glycol. Next, please. And these are the widely used polymers in biomedical field. This is polyparadioxanone. This is L-lactide also. You may all familiar about this. And this is another polyethylene glycol group biocompatible polymer. And our objective here to synthesize one new type of copolymer combining these three properties. And another, transformation of individual homopolymers properties to the conjugate product. And last one, improved biodegradable material also. Next, please. This is the copolymer chain which we synthesize in the laboratory. Here, the central part from the polyethylene glycol and the side part paradioxanone and L-lactide. Here also paradioxanone, L-lactide. The sequence, whether these are this here or this part here, this is random, because during the synthesis of this polymer, we mix this together with the polyethylene glycol. And polyethylene glycol we use dihydroxyl type, and these two hydroxyl groups have equal reactivity for the polymerization. So we believe, and some results also show that the block is structured like this way. The central part polyethylene glycol, which is water soluble, and side part hydrophobic, derived from paradioxanone and lactide. And for this study, we use these three different types of composition. First, paradioxanone and lactide ratio, like this, and the polyethylene glycol, one sample, molecular weight some higher, please. Next slide, please. And thus obtain this material we use as nanoparticles as a filling, and another is electrospool membrane. Just before presentation, some introductory results of this copolymer also mentioned about this electrospool membrane. Next slide, please. This is the one simple idea, simple escape to obtain the nano dispersion. Here, for this purpose, I selected the tri-block copolymer. First one, please. Previous. This copolymer here, polyethylene glycol is composition higher, and rest hydrophobic group is small. This copolymer was selected for the preparation of nanodispersion here. And first, that copolymer, this part is hydrophobic, dissolved in acetonitride solution. This copolymer is an antiphilic structure copolymer, since both hydrophilic and hydrophobic group here, and is dissolved in the organic solvent, transferred to the aqueous media under stirring condition, and is under the slightly vacuum condition, organic solvent evaporate out, and remaining copolymer as a dispersion. And this is the micellar aggregation, this type of particle obtained here. Next, please. And these particles were measured under the electrophoresis light scattering method, and the particle size and the potential here. And this A type, this is the composition, and B type, another composition. And here we can see L-lactide composition and this hydrophobic group composition increase. The surface charge, the zeta potential negatively increase like this, and we here, and the average particle size in this range. Next, please. and thus obtained this nanodispersion were applied for the in vitro gene delivery purpose. And these particles here, actually this part, this is not in my topics. I did only the polymerization synthesis part here. And this particle, from this photograph you can see, and other results also, this transcription efficiency was facilitated by using this type of nanoparticle dispersion. Here this blue color which is the representation of the lactate gene expression on this type of monolayer cell. This control and this particles. Next slide please. And here one results from previous slide that how much number of the cell transfected using this type of nanodispersion here. And this is the composition of the volume of nanoparticle solution with fixed volume of DNA, and here the number of lactate gene expression. This. Next. And next part is previous one is here the figure is some disappeared, but this figure electrospinning simple process. Just before presentation discussed about this type electrospinning process and the physics of this electrospinning process. You may already familiar and is every well known. Polymer solution kept here, and using the electric field, this polymer ejected out from the tip capillary as fiber, and these are some factors which can affect the morphology and diameter of the fiber collected here. This first one is solution properties, whereas viscosity and surface tension. Also, another affecting factor is the electric potential, concentration, and co-solvent composition. Also, sometimes we use this type of co-solvent. And also, another affecting factor is distance between capillary and collection is between here to here. And another one also, the motion of target is whether the rotation or the fix of this target may also. This also affects the morphology of the fiber collected. Next slide please. And this is the result obtained from electrospinning of the triblock copolymer. We use two types of solvent, methylene chloride and DMF, in different compositions, and the concentration of the copolymer was fixed. This condition for the electrospinning. Here we can see the process, how the varies from electrospinning to electrospray. The viscosity, depending on the viscosity of the solution here, the electrospinning, this side fine fibers, but this time the lightly spray the particle. And here, this is the fiber diameter of this photograph. You can see the less than 100 nanometer, the average fiber diameter about this was obtained. And this membrane, triblock copolymer electrospoon membrane, this is the result from the mercury porosity analysis, porosimetry analysis, porosity. Here the membrane thickness was used 0.5 millimeter and porosity this much, and this is the results from this instrument, and the distribution of pore size around this range, the rough range. And this porous membrane, the electrospoon membrane, was applied for the cell culture substrate. Next slide, please. And we use this membrane, and this is the procedure. You already well known about this. This cell culture, non-open fibrous structure was cut in this size and sterilized, and cell seeding is selected for the cell, this type of cell, and incubated here, and cell fixing. These are the methods, and at last cell attachment and the cell spreading. Here we saw by same. Next slide, please. This is the results obtained during the cell staging on the fiber matrix surface, and these different days. And on these days, after 10 days, overgrowth on the fiber surface was observed. Next slide, please. Here, some interaction of cell with the fibers, the cell growing on the fiber matrix here, and inside the matrix cell, and on the surface, and here attachment with the fiber, like this. These results, which I have mentioned here, is undergoing project and not optimized yet. Next slide, please. Yeah, this electrospool membrane was furthermore applied for the hydrolytic degradation, and both the film and fiber membrane was used. This is the phosphate buffer solution. This concentration is 7.5. The fiber was applied for the degradation at 37 degrees centigrade, and in different time interval, the fiber was with distilled water with washed, vacuum dried, and at last analysis. This is some results phenotypic observation. And first, here the composition polyethylene glycol some higher, and the film initially was translucent film and changed to the milky substance within 1 week. And it was difficult to see the morphology of this film. So we centrifuged and the freeze drying without methylene chloride, and at last the waxy material was obtained within 1 week. And but here the L-lactide composition at Faraday was some higher here, and film was translucent initially. After 6 weeks became white, and the very fragile 9 weeks, and sticky within the 14 weeks. Here this sticky material was low molecular weight, and rather there is no the Faraday only a lactide and polyethylene glycol copolymer, more or less the similar tendency here also. Next slide, please. This is the polarized microscope photographs about the spherulites, this film during the different types of degradation period. Here, no paradiagram, only the PET and L-lactide. The number of spherulites increased with growing size in this degradation time, and here the paradiagram applied. The number of spherulites rapid increase and excess, and size also increase, but here this size broken spherulite size. Next. This is the film surface observed in the same. This type of cracks were observed, and in the degradation time like this. And this is the electrospool fiber applied for the hydrolytic degradation. And here also you can see these bones, joints of the fiber is broken here, like this degradation photographs taken from sea. And this is also the enzyme degradation. The degradation process was carried out in freeze buffer solution. pH was 8.6, and this was selected proteinase K as enzyme, and here the same process as in the hydrolytic degradation phosphate buffer solution. And here the degradation was so rapid, within the 29 hours the degradation almost passed. So rapid. Next slide, please. This is some evidence about the hydrolytic degradation obtained from the carbon NMR. This is the control sample. Here, after 9 weeks, the new peak appeared within this range, which is from the previous reported results that this may result from the hydroxylated lactide in the units. It means in the degradation time, the small molecular weight fragments appear, and these appear in this spectra here because of this reason. Next slide, please. This is the proton NMR spectra. Here, this degradation time. So new peaks here appear, and here also new peak, and the number of peaks increase here. This type of number of peaks increase, and also the proton NMR spectra. The ratio of exactly this is the L-lactide and PEG only was considered. The ratio of these two parts also increase in the degradation time, which also shows the hydrolytic degradation of the copolymer. Next slide, please. This is some evidences from the differential scanning calorimetry. Some thermograms you can see here in the different degradation time. The position. This is the exothermic and endothermic position of exotherms shifted downwards, low temperature. Also, this is the second heating, first making and rapid quench, and furthermore heating. This second heating. Here also you can see the trend in within the degradation time. Next slide, please. This is some results from DSC thermogram previous. This here the crystallization temperature versus the degradation time, heat of fusion versus the degradation time. You can see crystallization temperature decrease, not so evidence, but decrease slightly, yes, and the heat of fusion increase. And the second heating thermogram, the results also the similar like before, but here the crystallization temperature decrease somewhat rapid than here. Next slide, please. And this is the weight loss and pH change within the degradation time about the copolymer film. This is the molecular weight increase. Here within 14 weeks the molecular weight MW decreased from this amount to about 7,000, 10,000 around, like this. Next slide, please. This is for the electrostone fiber membrane weight loss versus the degradation time. Water absorption was higher, about 250. It was different than the film because the electrostone membrane porous structure and absorbed water this much amount. But the degradation time, the pH was not so decreased, only up to this from 7.5 to 90. It's undergoing further for degradation. Next slide, please. This is this nanoparticle proposed. We use that polymer and change in molecular weight and the degradation time, and also the molecular weight pH here. Some evidence of the hydrolytic degradation here. Next slide, please. And this is the Watson spectra in different time interval of the degradation. After 7 weeks, the characteristic peaks corresponding to black time. Some evidence is here, but here almost the similar type. Next, this is the conclusion part here. Block copolymers of paradioxanone and lactide PEG has been synthesized and then furthermore processed as nanoparticle and electrostone fiber. And also the in vitro DNA transcription has been facilitated using the copolymer nanoparticle. Cell culture has been successfully done also on the electrostone membrane, and at last hydrolytic degradation has been effectively observed for the copolymer both in film and electrostone membrane. Next slide, please. And for this work, people we are doing good, and I would like to acknowledge. Thank you very much.